Hello friends, in this video lecture we will talk about uh, this is the second part of the genetic code video lecture. In this video lecture we will be talking about salient features of genetic code. What are the salient features of genetic code? In our first lecture we learned about what are genetic codes are all about, and what are the evidences that genetic code is triplet in nature. So regarding the salient features, the first character is its triplet nature. In fact, that is already being proved or already being seen in our previous lecture, that genetic code comprises of 3 nucleotides. And there are so many proofs or evidences for that. The first one is the mathematical one, the second one is Cram-Shift mutation, and the triplet binding assay. All these things prove, and nowadays a lot of things has been known. Sequencing is possible since 1977, of course, and after that it was very clear that genetic code is triplet in nature. Genetic code is non-overlapping also. That means if this is the sequence, then genetic code will be like this. This is one genetic code, this is another, this is another, this is the next one, like this. So there are 4 codons here, and many more next will be followed later on after this. So these are this way the codons are present. They are never coded like this, the next one is this, the next one is this. This will be called overlapping. So this does not happen. This is because a tRNA has to bind here, another tRNA has to come and bind here, and there will be 2 amino acids in the 2 sequences. They will be covalently tagged or covalently attached with one another. Then the ribosome will shift further, isn't it? So this is the thing what happens during protein synthesis, that tRNA having the anticodons, they have to bind, they have to align side by side, and so it is not possible for tRNA to overlap one above the other. That is why genetic code is non-overlapping. So the next thing is the character is genetic code is commaless. That means there is no punctuation. Here I can take the same sequence here, the ATG. If there is a pause or a punctuation here, that this next codon will start from, say, here, so this will act as a punctuation. This is not the case. Actually, this is continuous. Genetic code is commaless. It is actually continuous. Each of the 3 nucleotide sequences present one after the other they are coded by one or the other amino acids for what they are supposed to. So genetic code is without punctuation. Genetic code is degenerate. That means, because we have seen that there are 64 genetic codes, out of which 3 are stop codons. So there are next 61 codons that will code for 20 amino acids. So it is quite clear that there are more number of genetic codes than the number of required amino acids to be coded. So the system has given, the evolution has given this capacity that more than one codon are assigned for coding for one amino acid in certain cases, and that is not always that is not also equal in proportion. Some of the amino acids, like the methionine, is coded by a single codon AUG. Tryptophan is also coded by one codon. Isoleucine is ILE is coded by 3 genetic codes. There are others which are coded by 2, like glutamic acid or aspartic acid, and there are some other like the proline or glycine or valine. They are coded by 4 codons, and there are others like lysine, maybe serine. They are coded by 6 codons. In fact, so this is the variation that exists, but the 61 codons are altogether engaged in coding one or the other amino acid. So the cases where more than one codons are assigned for coding one amino acid, there exists a codon degeneracy. What is that? The first two bases in a codon, say if this is say I'm taking the example of phenylalanine, where in mRNA the sequence is U, U, U and U, U, A. These two sequences, they both will code, U, U, C. These two sequences, both of them will code for phenylalanine. So you can see the first two bases are specific. The last base is non-specific. And in fact, the cases where you see there are four, let me see the Codon Dictionary. Let me show you the Codon Dictionary first. That will make the thing more clear. Here is the Codon Dictionary. Here you can see this arginine. This is coded by four here and 2 codons here. So you can see in these 4 codons, the first 2 bases are similar. They do not change. The third base only changes. So the binding of a particular codon with the anticodon is such that, since this is the tRNA with this and 5' end and this and 3' end, this will be 5' end of the mRNA. So these are the anticodons. So if this is the sequence, say CGU, the sequence in the anticodon will be A, C, G. So first 2 sequences, this one and this one, the first 2 of the codon and last 2 of the anticodon are very specific binding. But the last one, it may be replaced by U, C, A or G. And this same tRNA will be able to bind, and probably there will be more than 2 tRNAs, more than 1 tRNA will be required in this case, because we will see how codon degeneracy also operates. It has also limitations that A cannot bind with all of them. A is very, very specific. In fact, A binds with U only. That means there is another tRNA. Actually, there are 32 tRNAs in the cell, about 32. It is being not clearly known exactly, but more than 32 tRNAs are present in the cell. They code for, they bring 20 amino acids as the amino acyl tRNA. That means there are more number of tRNAs for 20 amino acids. 12 extra tRNAs are there. What they are doing? They are actually for this codon degeneracy purpose, to read extra codons. And this is probably because to absorb point mutations in the system. That will be dealt in some other lecture later on. So we can see that the third base of the codon binds with the first base of the anticodon non-specifically. This kind of binding is called Wobble base pairing. This has been explained as Wobble hypothesis by Crick, because the A and the first base of the anticodon and the last base of the codon, they wobble together. They are not very specific, and so they can be altered. Say here A is very, very specific with U. That means there is another tRNA, maybe 1 or even more, that might bind with C, A or G. What is, there are options like that. There may be I in the position, inosine, at this place. That might bind with, I has option of binding U, C, A. I can bind with U, C, U or C or A. So it can bind with U or C or A, but not with G. That means there is another, there are 3 tRNAs are there for arginine. It is clear from this picture that there must be another for binding with the G. And in fact, there are other, 2 other bases, codons are there that will further demand presence of tRNA in the system. So out of these 32 tRNAs, most of them are being for arginine. It is not true. The proportion might vary, but it may be also like that. I cannot say at this moment then how many tRNAs are exactly there for arginine. Similarly, there are serine, lysine, and many other amino acids that are coded by more than one codons. So codon degeneracy is a common thing that is probably because probably it is there to absorb point mutations. If mutation occurs in the third position, it will rather be absorbed. It will not be. It will be a silent mutation. So the anticodons that hybridize with the codon U, if the anticodon contains U, it may bind with A or G in the codon in the third position. If it is C, it will bind with G. If it is A, it will specifically bind with U. If it is I, it will specifically bind with U, C, or A. So these are the options present. So tRNA contains so many derivatives of bases or so many uncommon bases like this inosine or dehydrouridine, DHU, or pseudouridine kind of thing. So tRNA, the anticodon portion of the tRNA contains inosine, and that inosine can bind with any of these three bases if they are present in the third position of the codon. So this is what is called codon degeneracy or bubble hypothesis. The codons are non-ambiguous. That means a particular codon always codes for the same amino acids. That means if U is assigned to code for phenylalanine, it will never code for any other amino acid. Like the UUC, if it is also for phenylalanine, it will never code for. This is called non-ambiguousness. It is never ambiguous. If it had been so, there will have been a lot of problems in protein sequences, and the system would collapse, basically. The next character is universal character, universality. The genetic code is normally universal, but there are exceptions. Some of the genetic codes, like you can say the AUG, that codes for methionine in the cell. AUG is probably, it codes for in the mitochondria, it codes for some other amino acids. Similar examples are there. We can cite like say AAA. It codes for lysine in the so it is a universal code for lysine, but it codes for asparagine in mitochondrial genome. Similarly, I will take the example. Say UAA is a stop codon normally, but it codes for tyrosine in the mitochondria. And not only mitochondria, but there are some other organisms also, like in the protozoa, you may find different kinds of variations. But 99%, rather 90, a little less, 98% cases, the genetic code is universal in nature. Codon biasness. So this is the characteristic. So for one amino acid, like the arginine, we saw that there are 6 codons. But is it that all the 6 codons are equally used for protein synthesis? There are equal proportion of tRNAs present for that? No. There is biasness. A few of the codons are used more than the others. This is called codon biasness. So these are the characteristics of genetic code. In our next lecture, we will see how the genetic code was in fact deciphered or cracked. Thanks a lot for your silent listening. This is all about